Okay, so we're going to do some more on nucleic acids. We're going to have a look at how DNA actually replicates. So if you remember, DNA is a double-stranded molecule, and each strand has these bases, and they form complementary base pairs. Okay. Now, if we have a look at just one nucleotide for a minute, each nucleotide is composed of a sugar. So you've got ribose or deoxyribose. On carbon 1, you have a nitrogenous base or N-containing base. Now that is the only thing that changes. And then up here, remember, ribose has a 5th carbon outside of the ring structure, and attached to that is a phosphate group. Now most of the time when we draw this, we don't draw that 5th carbon. And so if you drew that going out there, or going out, well, if you drew it going straight out, that wouldn't be correct. You have to draw it going straight up to suggest that there is that 5th carbon. So we have a sugar, ribose or deoxyribose, we have phosphate, and we have an N-containing base. This is a glycosidic bond, this is an ester bond. And we've come across ester bonds and glycosidic bonds before. Now, as I said, this is the only thing that changes. It can be adenine, which is an A, it can be thymine, which is T, it can be guanine, which is G, and it can be cytosine, which is a C. Okay, so that's the basics of it. They're the only bits that change. The sugar and the phosphate stays exactly the same. Now, if we have a look at these bases a little bit more closely, there are 2 types, and you can see I've capitalised some of them and lowercase some of them others. So A and G are one, and then the C and the T are the other. Now, there are 2 different types of these, and when you look at the structure of them, they have either a single ring structure or a double ring structure. I'm just going to draw them as circles because honestly they're more complicated than you need to know. Double ring structures are called purines. Can't spell. So purines are double. Now the way to remember that is that purine and double both have a U in. The single ring structures are called pyrimidines. Now the way I remember that, and I do know the difference between a 1 and an I, but the way I remember that is that pyrimidine has an I in it which looks like a 1, so it's a single structure. The purines has a U in it, and so does double. Okay? Now, you also need to remember which of these are the pyrimidines and which of these are the purines. The pyrimidines one is the more easy one to remember. Pyrimidine has a Y in it, so does thymine and cytosine. So thymine and cytosine, they are the pyrimidines. And I really can't spell. Pyrimidines. Yeah. And the A and G, they are the purines. Now, when these complementarily based pair, a purine always pairs with a pyrimidine. A always pairs with T, and C always pairs with G. The other thing you need to know is how many bonds. So A to T is 2, C to G is 3. I know I've said that deliberately. So A to T is 2, C to G is 3. And so the ones with the Y's in are the pyrimidines, the others are the purines. The purines are the double structures, the pyrimidines are the single ring structures. Okay, so let's have a look at how they actually replicate. How DNA replicates. So DNA is going to replicate when the cell wants to divide. When the cell wants to divide in mitosis, for growth, repair, whatever, it needs more of everything. It needs more organelles, it needs more DNA as well. So it needs double the amount so that when it splits, each new daughter cell gets an equal quantity. So if you've got a parent cell with some chromosomes, we'll draw 2 chromosomes, it has to double that DNA first. So then when you split it, each cell gets a copy of the DNA. Okay, so that's our copied one. You want to make sure, so they are both going to be the same as that one. So you have to replicate the DNA first, so then when you split it, they both get a long one and they both get a short one, for example. So let's have a look at how DNA actually replicates. Now, these bases are held together by hydrogen bonds. So one strand of DNA has the phosphate, sugar, and nitrogenous base. So that would be one. So we just draw it like that. That's the base, that's the phosphate and the sugar. So what you end up with is this backbone. It's kind of like the sides of a ladder. So this backbone is the sugar and the phosphate, sugar, phosphate, sugar, phosphate. These bits that stick out there, the nucleotides, they're the A's, the T's, the C's, and the G's. And so if you look at 2 strands, or one molecule of DNA, they're held together by hydrogen bonds. Okay? So, first of all, your DNA will separate. Now that is done by an enzyme called helicase. So helicase unwinds the DNA, because remember it's a twisted helix. So it unwinds it so you get this kind of ladder kind of a thing, and it breaks the hydrogen bonds, exposing these bases. Now inside the nucleus you have some free nucleotides. They're just nucleotides that are kind of ready there for DNA replication. So they will come in and they will line up opposite their complementary base. And then the phosphodiester bond that joins these nucleotides to each other, they are formed by polymerase, DNA polymerase. So DNA polymerase will add in each base each time, knitting that phosphate sugar backbone together. Okay, and so you will start to get your DNA elongating. And the same thing happens on the other side. I'll do a bit more detail on the other side in a minute. So you just put your bases in. DNA polymerase knitting that phosphate sugar backbone each time, and you end up with 2 strands of DNA. Okay, so you've got 2 enzymes, you've got helicase, you've got DNA polymerase. Now that's the basics. That's what you absolutely have to know. So what I'm going to do in the next video is go into much more detail about these 2 different processes.